Hello all, this is Menaka Thambiraja from Sastra Deemed University, Tamil Nadu, India. My research entitled is Genome-Wide Structural Variation Analysis of Indicine Cattle via Whole Genome Sequencing. The major focus of my research is the comparative genomic analysis of copy number variation among Indian, exotic and crossbred cattle using whole genome sequencing approach. This research particularly focuses on identifying key genes that could responsible for disease-resistant trait in Indian cattle. This because of the Indian cattle have inherent features such as adaptation to tropical climatic condition and resistant to various infection which compared to European cattle. This advantage of Indian cattle because of their evolution under specific agroclimatic condition. So we are particularly interested to identify the unique innate immunity genomic signature in Indian cattle. This could enhance the immune competence and resistance capability of Indian cattle. This therefore lead to the improvement of breeding decision and development in future endeavors. The workflow of our analysis is whole genome sequencing of Indian cattle, which is 4 Indian cattle. They are Kangayam, Gir, Sahiwal and Tarparkar, and 1 exotic cattle which is Holstein Friesian, and 1 crossbred which is Karen Fries from 18 different locations. Then we perform 2 different analyses. The one analysis is identifying CNVs in genes using the Delhi tool. These genes commonly present in all 6 cattle. Then we identify the immune genes from those genes. Then we identify protein-protein interaction of immune genes using string database. Then we perform docking of immune complex using HADDOC software, and finally we perform protein-protein interaction analysis of docked complex using PDB sound tool. So another part of analysis is to identifying the breed-specific CNVs in innate immune genes using MANTA tool. Then we identify the significant variation that present in innate immune genes of all Indian cattle but that were absent in exotic and crossbred. Then coming into the result and discussion part, so this is the CNV analysis with Delhi tool. So these analyses reveal 149 genes that have a copy number variation which include break-ins, deletion, duplication, insertion and inversion. So these genes commonly present in all 6 cattle. From this we identify 42 immune genes including innate and adaptive immune genes that truly based on literature survey. And then from this we extracted 8 innate immune genes that compared with innate DP database. So here I have mentioned the 8 different innate immune genes. So then we perform the protein-protein interaction analysis of those experimentally reported 42 innate immune genes. So here in this table I have mentioned the immune genes that have experimentally exhibiting more than 10 interacting partner as the confident cutoff of 0.4. So this network reveals that the protein-protein interaction network of immune genes with more than 10 interacting immune partners. So the violet color indicate the immune genes and orange color indicate their interacting partners. So this table indicates the docking analysis of experimentally reported those 42 genes with their interacting partners. So overall we performed 112 docking for those listed innate immune genes. So here I just represent the one complex which is BCL2 and their interacting partners BK1. So first we generated the protein structure of immune genes and their corresponding interacting partners using AlphaFold. So then we perform the docking analysis using HADDOC software, and then we identify the interface statistic of docked complexes using PDB sound tool. So here I have mentioned the protein-protein interaction analysis of docked complex which is BCL2 and BCL2 BAC1 complex. So the interface statistic we obtained from PDB sound tool. So here I have mentioned the docked complex of innate immune genes with other innate immune partners. So further I have mentioned this table as protein-protein interaction analysis that reveal the interacting bond between the immune protein complexes. So the complex have a higher bond which means most stable interaction. So here I have mentioned the docked complex of immune genes with their most stable interacting partners. So this is the another analysis, which is breed specific copy number variation present in innate immune genes with structural variation. So the upset plot revealed the common innate immune gene which present in all indigenous cattle that have a ingestion variation. Here you can see around 27 immune genes, particularly innate immune genes, have a ingestion variation that commonly present in 4 different Indian cattle. So here I have mentioned the detailed version of those 27 innate immune genes. So for example, here I have mentioned the genes and their corresponding structural variation length of insertion, and in the bracket I have mentioned the number of insertion events that undergone that particular gene. So this table indicates that the common genes that present in all 4 Indian cattle but that were absent in exotic and crossbreed. So this table indicates that the impact of duplication which is present in innate immune genes of Indian cattle, exotic and crossbreed. This is the same upset plot for deletion variation. So in overall, we identify the 19 immune genes that undergo insertion variation and 9 immune genes that undergo deletion variation that present in all Indian cattle, but that were absent in exotic and crossbreed. So these significant genomic differences we could identify using this meta tool, and the duplication that present in 2 immune genes in Thar Parker and 6 immune genes in Sahiwal cattle. In overall, we conclude using the Delhi tool, we identified the 8 innate immune genes that have a genomic structural variation, and through protein-protein interaction analysis using string database, we identified the immune genes that have exhibiting more than 10 interacting partner, and we also identified the innate immune genes with other innate immune interacting partners. As well, the BDB sum analysis revealed that the most stable interacting complexes between the immune genes, and further we also see the significant genomic differences which present in Indian cattle but that were absent in exotic and crossbreed. So I could acknowledge the ICR NASF for funding and Shastra deemed university for infrastructure support and research facilities, and I welcome Gene ISCB for this opportunity. Thank you.